Hello everyone, my name is Christian Jachnovic. I work as EATG Training Coordinator for the EATG, and I'm happy to welcome you to our webinar on Introduction to Medicine Research and Development. We have two great speakers today with us. Tamás Bereczky will talk first, and he will talk about the basic principles of medicine discovery and development. And following Tamás' presentation, Giulio will talk about the role of community in the process of medicine development. Before Tamás starts, I just want to give you a short introduction to on how to use the GoToWebinar. You all see a panel which allows you to write questions. So there is a question window where you can put your questions in writing. I will monitor the questions and then ask them to the speakers. Or if you would like to become unmuted, because at the moment you are all muted by default, but if you would like to have your microphone switched on, you can raise your hand. There's a small hand button, and then I will see it and unmute you. So that's all from my side. Tamás, I am making you presenter.
Thank you. So welcome everyone, and I hope you can see my screen, which is the title screen. Can you confirm, Christian, that it's on the screen?
Yes, I can see it.
Great, actually. Brilliant. So welcome everyone. My name is Tamás Bereczky, and I work with the EATG and also with the European Patients Academy on Therapeutic Innovation. That you can see, EUPATI is the abbreviation, or is the name, which is well, it is what it is. It's a patients academy. It's a school for patients and patient experts and people who want to learn more about clinical research and development, or biomedical research and development and clinical trials. I'm a patient myself. I have been living with HIV for 15 years now, and have been working as a peer helper and educator and a patient advocate for the last 12 years or so, maybe 13. These are my disclosures. I receive no funding for my research work. And I would like to, first of all, say a few words of acknowledgement to these people on the screen, who partly developed the following slides and also who have done a lot in order to make EUPATI and our work successful, Giulio included, who will then speak later and explain some aspects of biomedical research and development from the patient's perspective and patient participation perspective. Now, I have tons of slides now, so I will rush through this collection of slides relatively quickly. If you feel that something is unclear or something that you don't understand or want to, you need clarification, just as Christian explained, let us know, and then I will try to explain that. Otherwise, I will just throttle on at full steam because it really is a mouthful, as you will see. Basically, what I will outline to you is how the whole process of medicine development is built up, how it is structured, and as you will see, this is a tremendously complicated and time and money intensive process, which is absolutely necessary to understand how these different building blocks work together in order to be able to position yourselves in this process as patient advocates if you like, lay people coming in from the outside, yet experts in this process. And this is exactly what then Giulio will explain in more detail. Now, on average, it's 12 years, and according to some statistics, about €1 billion that it takes to do the research and development before a drug can reach the market. So it really is a very risky venture and a very risky investment and work. And we also know that only 2% of the leads or of the compounds that we start, or that companies start working with, will reach the market. So 98% is eliminated during this process, during these, say, 12 years. There's a lot of work going on to shorten this period and to reduce the costs, and there's also some uncertainty about how much the real cost is, or how much it should be, but that's a different discussion, and that's not something that we will talk about much today. What I will outline to you is more how this process looks like today, and what the most common processes are in biomedical development or in medicines research and development and clinical trials. So if you look at this whole process, how it is built up, you can see that there are 4 distinct phases that I will touch upon today. The first is research and discovery, and then the next is non-clinical development. Then comes clinical development. This is what we talk about the most. This is where you will find the clinical trials, phase 1, phase 2, phase 3 clinical trials. And then there's also a 4th stage, which is the post-approval stage, or we also call it lifecycle management and pharmacovigilance. Pharmacovigilance is the process, or is the concept, when the actual use and existence and long-term consequences of the use of a certain medicine are monitored on the market. On this chart, you can also see the time requirement of the various stages of research and development. And then Giulio will talk in more detail about how patients can get involved in these various stages. But right now, I will go through these stages together with you, and I will explain what happens in each of these stages. So let's start with the first one, which is the discovery stage. First of all, before discovery, before a certain medicine is being conceived or thought about, we need to determine whether there is an unmet medical need for that given area. This largely happens in basic science. This happens in academia, in universities or research institutions, and also to some extent in the industry, in pharmaceutical companies, where they try to understand how certain illnesses, how diseases work, and how they are present in populations, and what the unmet need actually is. So the unmet need can be that there is no suitable or no effective medicine at all. We still have a lot of illnesses, not only rare diseases, but also more common diseases where there's no medicine, there's no cure. Actually, there's very few cures for medicines. Most of illnesses are manageable, but not necessarily curable. Or the unmet need can also mean that there is something available already, but some patients cannot take it, or they are resistant to some treatment, or there are unacceptable side effects that makes it necessary for research to come up with new solutions. Now, the next step is, and of course, I forgot to say, but you will get all the slides then, so everything will be shared with you, so that you can follow up on this again. So the next phase in discovery is actually when we try to understand what is the unmet need and how this unmet need can be covered. There's a huge array of medicines or of illnesses and conditions where medicines are still not there, so where there's still not development has not even started, and also already in this stage, there is a regulatory process that needs to start. So in addition to the scientific work, there's also an administrative legal work that needs to start and needs to be done in order to make sure that then the medicine, when it is developed, can reach the market. Now, there are different new models to this, and we have been talking about this within the EATG, but also within EUPATI about various new models of pharmaceutical development. This is not the topic of our talk or of our webinar today, but you should know that this classical paradigm that how clinical trials and how clinical research is built up is currently being shaken up a little by various alternative paradigms and attempts. Now, the next step is that this whole process needs to be done in a very structured phase. This means that, of course, this is also a business case. This is also a business decision. So it's not only about the unmet medical need, but it's also about the business aspects or perspectives of the given treatment or of the given development. And this is what then leads to, so this consideration leads to a so-called investment decision. The investment decision is basically an agreement for the amount of money that needs to be made and the amount of people or the human resources, knowledge, if you like, skill sets that need to be allocated to do the work. This, what we call the investment decision, is then taken within the company, or it is taken within the research institution or the university, which does this type of research. So you will see that this whole pattern of considering first the theoretical background, then doing an activity, assessing the results of this activity, and then taking another decision. So this step-by-step stage characterizes the entire process. And this is which also allows researchers to interrupt research whenever it turns out that the given compound or the given procedure is not suitable for further development. Now, the next step is what we call target selection. Target selection is explained in very simple terms, is when you try to define that part, that bit of the illness or of the pathogen that causes the illness, where you can attack it and where you can make a meaningful intervention and change. Because we know that disease occurs when normal processes in the body are disrupted or are altered and there's no proper functioning anymore. And we have to understand in detail, and in detail means that really on the level of cells, or increasingly on the level of molecules and genes, what has gone wrong. And then this abnormal process needs to be targeted. Now, this target, as you can see also in the slide, can be a molecule of which there's too much, or which interferes with normal body functioning, or maybe there's not enough of that given molecule, or maybe that given molecule has an abnormal structure which needs to be changed in order to overcome or in order to manage the given illness. So if you look at this example, and this example is from the cancer field, and I will also speak a little about HIV then, you see that in this case, there can be various targets that you can select. So maybe you can select the growth factor if it is about cancer. So you see that this growth factor will affect the nucleus of the cell, and this causes excessive growth of the cell, which leads to cancer, right? So a tumor is too many cells growing in an uncontrolled way and not dying off at the right time. So maybe if we can target the growth factor, or if we can target the growth factor receptor, or maybe if we can target how this message coming from the growth factor is conveyed into the nucleus of the cell, then we can stop this process. And this is where the target selection, this is what the target selection means, that you try to find out where you can make the easiest, the most effective intervention into this process. So as you can see, this example came from cancer, or comes from cancer, and on this slide it is then described in more detail. So there's a chemical messenger that combines with the growth factor, a receptor on the cell surface, and this is what generates a message inside the cell. And this uncontrolled growth leads to cancer, as I already said before. And if you can block this receptor, then this means that this uncontrolled cell growth will stop. And this means that the given tumor, the cancerous cells will no longer grow. Now, oftentimes, this is an extremely complicated process, and it's impossible to say precisely which abnormality or which target is responsible for the given illness. So this means that there must be several attempts in order to get to the right target. And also, it is possible for this reason that we select the wrong target. So therefore, finding and selecting the best target is crucial for success. Today, this process happens primarily what we call in silico, so in computers, through computer modeling, and to a lesser extent in petri dishes, in laboratories. But largely, there are vast collections of, or libraries, if you like, of molecules and of various methods and pathways in companies and research institutions, which can then be modeled through computer modeling to better understand where the suitable targets for a given illness are. Now, if you look at HIV, in order to make it more relevant for ourselves in the HIV community, then we can see that these targets in the field of HIV have already been mapped pretty well. Still, there are new targets, especially with personalized medicine and with gene technology and genetic engineering coming into the picture. We find that there can be new targets where we can perhaps intervene into the life cycle of the HIV virus. And this is exactly why cure research has become so essential for understanding how HIV works and how the whole life cycle of the HIV virus looks like, because it allows us to find perhaps new targets in the treatment and management, and then eventually also in the cure of HIV. So you see, this is a very early stage of the development process. Now, if we move on in this process, the next step is called lead generation. Lead generation means that you find one particular or a group of molecules, a small group of molecules that we know that can interact with the given target that we have selected. Now, these can be naturally occurring molecules, or these can be chemically, artificially manufactured molecules. These can be large or small. These can be proteins. Proteins are very complex sets of molecules, and then we call them biologics. And when we test these leads in order to understand whether they really do the work that they want them to do, is called the screening process. Right now, this is again done in silico, so by computers and in an automated way, so through robotic technology. High throughput screening is one of the methods that's being used, which allows that we can test millions of molecules in a relatively short time frame. And once you have selected this smaller group of molecules that we think, or proteins that we think that can do the job that is needed, then we can move on to the next step. And the next step is called the lead optimization. Now, the lead optimization means that, okay, we have found a molecule now that we know works well, but maybe its effect is very small or it's very weak. therefore, we need to understand how we can strengthen this. So you need to optimize, you need to tweak and tailor that given molecule or that given protein that we found in order to come closer to what we want to do. This example again comes from the cancer field, and why there are so many examples from the cancer field is because there's tremendous amount of development going on in that particular area of medicine right now. Unfortunately, some other illnesses are more neglected, but still there's a huge amount of cancer related illnesses that need urgent intervention. So there's a lot of work going on there, and that's why there are so many examples emerging from that field. So you see that chemists and biologists are able to tweak these molecules, adding here and there, adding parts or parts of the compound, elements or additional molecules, smaller ones, into the original lead in order to change its effect and to strengthen or weaken, amplify or tone it down as needed. Now, this lead optimization is not at all a very simple process, because then these molecules need to be tested again to determine which of the selected structures or of the structures developed best fit the purpose that we want to attain, that we want to reach. And then when we have done this, then we can lead this on to the next step, which is that we call them candidate medicines. So they become from a simple bunch of molecules, they first become leads, and then they become candidate medicines. We are still very far away from them becoming medicines, actual medicines or pharmaceuticals. So this is when the next important legal steps are already also taken, or regulatory steps, which means that these molecules, these compounds, these candidates are registered or patented and come under protection as intellectual property. Again, this is a process that's being challenged right now from also from patient communities, but also from research institutions, but we will not talk about that in detail. So this is when we already enter the non-clinical stage, or rather the end of the non-clinical stage, when it comes to safety testing. Safety testing is done in animals, either in humanized animal models, which means that these animals are slightly altered, changed, their genetic material is changed to make them more similar to humans, or their tissues are more similar to humans. Actually very expensive animals, and also this work is done, or already the whole process is done, and also this work is done according to what we call good laboratory process or practice rather, sorry, which describes how these studies need to be done and how this kind of work with animals needs to be done in order to be safe and also reliable. This is how these are the objectives of this work. So is this what is the effect of the medicine in the given organism, in the animal, in the different tissues and organs? So there are also toxicology studies that need to be done. Whether this allows the animals to reproduce and develop normally, or whether it inhibits them, so it's called reproductive toxicology. What's the effect on the skin? What's the effect on the eyes, on the level of chromosomes and genes? And even if these compounds are maybe carcinogenic, so whether they cause cancer or not. This slide describes the types of toxicology studies. I will not explain this in much detail. You can look at the slides then later. We are still at the non-clinical safety testing. So you see that this slide describes the what we call the ADME process. So we need to understand how the substance enters the body. So what's the absorption, how it is distributed around the body. Not every compound, not every molecule will enter every part of the body. There are compartments in the body. The most known of these is the blood-brain barrier in the humans, so that not every molecule can pass into your brain. And this is also true for animals. So we need to understand how molecules are distributed in the body, how they are broken down, so how the metabolism works with these substances that are given to the given subject or animal, and how it leaves the body, so whether it is excreted by the kidneys or by the liver. So all of this needs to be done and understood in order to see or understand or decide whether the candidate compound can then proceed to human trials. And that's where we transit into the clinical stage of clinical studies or of clinical trials, when it's already in humans. For this, you need a first approval from the national competent authorities, so from the regulatory agency. Also, you need an ethics approval. You will understand, or you will see later, that this is already a stage where patient representatives can play a major role. And safety is the most important aspect. So no study in humans can start before an approval from an internal, so a company internal review committee, or an institutional internal review committee, an external ethics committee, and also the external regulatory authority, which must give approval in order to give these compounds to, or give the trial compound to humans. Now, this is usually done with healthy volunteers. So it's the phase 1 clinical trial stage, which is done with volunteers, healthy people. This allows doctors and scientists to test if the medicine is safe in humans. And this is why we call them proof of mechanism studies. So we just look at whether they are safe to give. And how it behaves in the human body, whether it's similar to how it worked in animals. You will see as we proceed that at every single stage there's a huge chance for the given medicine to be abandoned, because it's not safe enough, because it's not effective enough, because it works in a different way than it did in animals, or because it already worked differently in animals than it did in the petri dishes. So this is, you see, so this is why there's a continuous monitoring process in order to understand what works or how it works in the human body. So you need informed consent, and also you need ethics committee review in order to be able to proceed further on in this. Informed consent refers to the participants, so they need to understand what is going to happen to them in the trial. So phase 1, we are still here at this stage where safety is our priority. We started with a very low dose, then comes dose escalation, which means that you increase the dose to understand what happens then. Then we analyze the study results, or the scientists analyze the study results, and they try to understand pharmacokinetics, which is what your body does to the medicine. So that's called pharmacokinetics or kinetics, and also pharmacodynamics, which means that we try to understand what the medicine does to the body. So it's pharmacokinetics is what the body does to the medicine, and pharmacodynamics is what the medicine does to the body. Both needs to be understood in order to be able to proceed to phase 2. And if we find that the phase 1 study results are good enough and it's safe to proceed, then the next step is to start with clinical trials in patients who actually have the given illness that needs to be treated. So we usually have two groups. These are the so-called placebo-controlled studies. So we have one group that receives the actual medicine, and another group that receives the medicine that has no effect on the body, which is called a placebo. There's again a lot of debate and a lot of discussion about using or not using placebo-controlled trials. These trials are already a little larger so it's they are carried out in 100 or 500 patients. This also depends on the availability of patients. And the main objective of these trials is to understand or to gather information about the effect of the given medicine on the actual illness. So this is already a proof of principle as opposed to a proof of mechanism trial. So it's a proof of principle trial, which means that we try to understand whether this given compound really does to the illness, to the ill body, what we wanted to achieve originally. So these phase 2 trials are already run in several hospital sites and several doctors. They are called investigators. Of course, this is a more complicated process than if you just have one single site. These trials are usually already outsourced to what we call a clinical research organization. A clinical research organization has a single job, which is to organize clinical trials. So it's not necessarily the same company as which develops the compound or the medicine, but it's an independent organizing, if you like, logistics company, which will take care of organizing the trial. So you see, it becomes even more complicated again. It usually takes something like 8.5 years, and the costs might run up to €1 million. And you will see that even at this stage, from 10 medicines that are tested in phase 1 and phase 2, on average, there are only 2 which will make it to the next phase. And the next phase is phase 3. Now, phase 3 trials are also called confirmatory trials or studies, where we try to understand what the given trial medicine does in terms of efficacy and safety in large patient populations. So this is where we reach already thousands of patients that receive the medicine. All the information from earlier stages is used to take decisions, including a final formulation of the medicine. So nothing is final still in phase 3, and trials and medicines can also still be stopped even in phase 3 if in larger populations scientists or researchers find out that the medicine is not safe enough, if or it's not efficacious or not effective enough. And because they also require a huge amount of expertise and large logistical work, this is why at this stage we always find clinical research organizations or CROs involved in this process, which take care of the organization of the work. Now, this is all very cumbersome, and this is all very, very difficult and expensive, but this is the only way to see in a scientifically structured way to get a clear answer about the efficacy of the medicine and its safety, right? So how well it works and how, if and how well it is tolerated. Over 50% of the medicines that reach phase 3 will still fail. And this is how we come to this 97–98% of total failure rate that I already mentioned in the beginning. I said 98% in the beginning, 97% is written here, but it's more or less in that range. So a large amount of the initial ideas must be abandoned during the process. And this is the argument that you will hear so often, that because this takes so long and because the risks are so high and because this is so expensive, so that's why it is also then that's why the medicines are so expensive. And that's there's a lot of debate about that. But as I said, this political aspect is not the subject of our discussion today. Now, the next step is regulatory submission. So you need to submit all of these documentations and all the data, all the raw data, which can run up to millions of lines of data entries, have to be submitted to the regulatory authorities. And then they, if they are satisfied with the results, and this, of course, again, takes a lot of time and a lot of work and effort from the regulatory agencies to understand. So if they are satisfied with what is called a dossier, then this is then the marketing approval is granted for the medicine. So you see, this is a tremendous amount of work behind all of this. It usually takes up to 1.5 years for the regulatory agencies. Primarily, we mean now the EMA, and Giulio has been working with the EMA now for quite some time, so he can give you more details about this type of work. And also, there is an additional step now which is coming, which has been there for the last 5 to 10 years, which is health technology assessment. And if you're more interested in health technology assessment, then there are several webinars that have been produced by EATG and by EUPATI about this particular step in the medicines development process. So we also must remember that after marketing, there's still a lot of work that needs to be done. So this is called the post-marketing safety surveillance or pharmacovigilance process, where in clinical trials and also on the basis of real-life data, we collect this information in order to understand the real risk-benefit relationship. And then life cycle management is where, or how we describe this final process, which means that we try to understand from the earliest phases until when the medicine has been on the market for a longer time, how it plays out on long term. This you will see that this is true for both prescription-only medicines, but also for over-the-counter medicines. So because all of these require approval from the regulatory authorities, so the same procedure, the same oversight, and the same monitoring is needed for all of these. So you will see that there's different types of clinical trials. Clinical trials, what I have described so far, clinical trials can be interventional. This is when you do an intervention to a patient population in order to understand how that intervention works, so what the outcome is. It's a classical experiment, right? So there is a patient population, they live with a certain illness, you make an intervention, so you add a new medicine, you administer a new medicine, and you see what the outcomes are. So it's a classical experiment, and you measure the outcomes. But clinical trials can also be observational. So there can be cohort studies when you look at a large group of patients and how their lives and how their numbers, how their data evolve over time, and you try to understand what that implies for the given illness and for the patient population. They can also be case control studies, which means that you look at individual patients and their numbers and their data, or they can also be cross-sectional studies, which means that you look at both at the same time through statistical modeling. And then there are pilots, and there are feasibility studies and screening trials, which is testing. Screening is testing in this case, or can also be described as testing. And then there are prevention trials. There's a lot of those going on in HIV prevention right now with the PrEP demonstration trials. And there can be epidemiological studies and sequential trials. But what we are talking about primarily now are interventional clinical trials that I have described to you so far. Now, you will hear from Giulio that patients play a key role in all of health-related research. So you will see that this is something that's been tremendously wide-ranging and important for the whole patient communities. And we also know that this whole process of clinical development requires and allows patient participation. But we also know that there's not enough of patient advocates and patient experts to be involved in this process. So the answer to this question, whether there are enough patient advocates, is no. And that's why we have EUPATI, that's why we have the Patient Academy, which has developed materials for patient experts, for patient advocates to understand this whole process better, and in order to be able to also make a meaningful contribution to it. This work has been described extensively, and is also reflected in the EUPATI Toolbox on medicines research and development. So if you need any additional information about what I have been talking about so far, then you just go to www.eupati.eu and you will find articles, PowerPoints, infographics, fact sheets, and even mini courses, starter kits, we call them, presentations about what patients can do and how they can get involved in clinical development. You can run whole training days through this, and you can reach me on this email address. And now I pass the floor to Giulio because I'm running out of time already. If you have any questions, I will have time to look at the questions now, and I will start answering them. I will pull up your slides, Giulio, now, and yes, that means that just hold on for 1 second.
Yeah, thank you.
And there you go. Your slides are up. You take the floor.
Okay. Thank you very much, Tamás. Just tell me if you can hear me. Okay, I suppose you can hear me. But,
yes.
Okay,
Yes, I
okay.
That's why I did that. Yes, but we can hear.
That's good. Okay. So welcome everyone. Thank you for attending this webinar. You can go to the next slide, please, Tamás. I just want to briefly introduce myself. I will not read through the slide because it's but I just want to highlight that I'm just a patient, a person living with HIV. I got involved in patient organization, and being involved in patient organizations, I had the chance, the opportunity to interact with various stages at various stages of the medicine development. So I am part of the European Community Advisory Board. I sit at the EMA representing the EATG. I'm in some trial steering committees, and I'm also a member of an ethics committee. This is just to show that anything I will discuss in my presentation comes from my own experience, so it may be not objective, not widely shareable, so to say, but it is also based on some information we got at the EUPATI. I am also a member of the EUPATI editorial board, so it's more or less something that can be shared among the community of patients. Next slide, please. So Tamás showed you a slightly different slide about medicine research and development. I choose this one, which highlight the various activities where patient representatives can be involved in the process of medicine development. So in the next slide, we can see a list of these activities, just a bit grouped together. We start from setting research priorities, then there is the whole process of medicine development in preclinical and clinical trial that Tamás brilliantly explained, the ethical review, and so on. The health technology assessment and the regulatory affairs. Let's try to see each of these steps a bit in detail. Next slide, please. So, setting research priorities. What does this mean? This means that at the moment where the researchers or the sponsors or the pharmaceutical companies are trying to identify the new medicine to be investigated, they can interact also with patients, trying to assess several questions. What are for example, the objective of the trial that must be ensured for it to be relevant for patients. The inclusion and exclusion criteria for trial participants, for example, are acceptable endpoints, and all that has to do with the trial design. Next slide, please. So usually this activity is done in an advisory board. The sponsor, the ones who are going to develop the medicine, can convene, can meet with different people, patients, but also usually also clinicians, to discuss for example, the therapeutic needs and priorities, and trying to identify which are the unmet medical needs that must be addressed. In this activity, of course, you must know more or less what are the therapeutic options which are available for your disease. In our cases, for HIV, for example, HIV infection, and how these medicines work. Also, this must be known. Quite delicate element is the fact that you are sitting at this table trying to bring not just your own personal experience, but you are there to represent the needs of the whole community of patients. So this must be done in a balanced way. Of course, we consider our priorities as big priorities, but sometimes they're not the ones that are considered as priorities for the whole community. And this raises a question which is quite relevant about who is the right patient who can engage in this discussion with sponsor researchers about setting research priorities. But I must say in the whole process of medicine development. So in the next slide, I'm quoting the definition of patient used within the EUPATI project. So we can identify different kind of patients. There is the individual patient, the one who is living with a disease. He or she may know very well what this means to live with that disease, but maybe he or she has no technical knowledge on research and development, no knowledge about regulatory processes, and so on. There are carers, those who take care of a person living with that disease. They can know very well what the disease is because they live with someone who is suffering from that disease. There can be patient advocates, who are those who try to support the population of patients living with that specific disease. And this may be done within a patient organization or not. It's a choice. Then there are the patient organization representatives, who are usually there to represent and express the views of the whole community of patients with that disease. And the patient experts, there are the patients who, besides the fact that they live with the disease, that they are able to represent also the need of a patient community. They have some technical knowledge on research development, or regulatory processes, all the stuff that are covered, for example, by the EUPATI process. This is just to say that in some cases, the individual patient can bring his own experience, and that's good, and that can be helpful in some processes. But usually, patient representatives are asked to bring not just their personal experience, but the experience of the whole community. So a good connection with that patient organization, for example, or at least in general with the community of patients living with that disease is needed, and it's quite important. Next slide. So I'm trying now to highlight some challenges and opportunities coming at each step of this process. When discussing with researchers about research priorities, we have the opportunity to contribute to significant advancement in the treatment of a disease, in HIV, for example, or in any disease, because we can express unmet needs and then try to advocate for a treatment option that could address them in a more proper way. And also, we can help in designing the whole research and development program in a way that which is more friendly, so to say, more closer to the patient's needs. But of course, there are some challenges. You are sitting there representing the needs of the whole community, so it puts you on some big responsibility, and also some knowledge in basic science can be of help in these cases. So next slide, please. Okay. So the next step is the protocol synopsis and the design of protocol. in this phase, of course, all the technicalities related to the protocol design. So how many participants are going to be involved to make that the trial being acceptable, but also representative and important for the research objective. The inclusion and exclusion criteria, the risks for trial participants, the burden of these are the risks, how many blood samples, for example, they must give, or so on. All these aspects, which can be considered technicalities of the protocol, can be designed in a way which is more friendly for trial participants, and this is our role as patient representatives there. So next slide. Usually the review of the protocol, the intervention on protocol design are done in a smaller group. I will give you briefly the example of our experience at ECAB. For example, we at ECAB have identified a group of reviewers who have more knowledgeable about protocol design and all the technicalities I was referring to earlier. Once a company asks for a protocol review, we can send out a call and identify 3 or 4 reviewers, and also identify one reviewer who will be coordinating the whole review. There is, of course, an issue related to confidentiality. So we at ECAB have a confidentiality agreement which usually covers this activity, but sometimes companies ask for a supplementary confidentiality agreement to be signed. Then the reviewers have some days, from 3 to 15, to review the document and send the comments. The coordinator will collect all these comments, put them together so that they are not connected with the person, with the name of the person who asked for the comment, for the question, or made that comment. And if there is some discussion about internally, we can have a TC just to identify, to solve some contradiction that may appear from the comment of one reviewer from another. And then the document is sent out to the company that usually organizes a call to discuss all the comments a few days later. Next slide, please. So at this point, I think that the opportunities are pretty clear. You have the opportunity to help in designing a study, a protocol, which is more friendly to trial participants. Also, you can identify some information which could be good to be collected during the study, and something that might be less relevant for the researchers and is more relevant for us as a community of patients. And also discuss the inclusion and exclusion criteria. I must say that usually in protocol review, the 2 parts which are more relevant for comments and discussion for us as patient representatives are exclusion and inclusion criteria, just to be sure that the sample population is somehow responding to the real life population which is going to use the medicine, and also, of course, all the parts related to safety which are really relevant. There are some challenges that you need to have some good knowledge on medicine development and trial conduct also to review a protocol. You must know what is available there, what are the data for that specific compound, also the preclinical data. So you must have a global knowledge of the field. And you also have to challenge the researchers' attitude, because they usually have this attitude to consider participants to a clinical trial as more or less guinea pigs, so to say. So it can be challenging sometimes to make them think of trial participants as people living with the disease, and try to consider, to respect also the contribution they give to the trial conduct. Next slide. So we go to the whole document related to the information. You know, what a medicine is developed at various stages, there can be some information that need to be sent out to the patient community, or also just to the trial participants. Now, researchers have their own language, so they usually have are not very clear for lay people in the language they use. So all the documents for information, includes the informed consent, must be written in an understandable way, and that's usually done by the contribution of patient representatives in this process. Sometimes this process can be done within the trial steering committee group. It happened to me sometimes. We will see what the trial steering committee is in a few slides. This process can be cumbersome when it comes to multinational pharma-sponsored trials, because they usually develop just some formats, some templates, but the actual informed consent form, or also the information for the patient community, are developed at the national level. So it's easier to get involved at that level if you want to contribute. Usually it is done by individual patients, but as I said, language and readability are the main issues here. Next slide. So the opportunity is, of course, to ensuring of the research. In this case, the challenges are that some communication skills are needed. You should be able to translate complex scientific information in lay language, and also to interact with language used by researchers. Next slide. Ethical review. You have some everything that has been discussed so far can be part of the ethical review made by patients. But usually within the ethics committee, there are some more issues that must be addressed. For example, data protection or conflicts of interest, but also the insurance or all these kind of technicalities. You can read it on the slide. Next slide, please. So this process usually happens at an ethics committee. Ethics committees are very different from country to country, but in most settings, you have a patient representatives at the ethics committee. And also the clinical trial regulation, which will come into force next year, states that at least one layperson shall participate in the assessment of the clinical trial application. So this will become a common rule, at least in the European Union. If you want to participate into an ethics committee, you can also sometimes spontaneously apply for this. That's what I did. I am part of an ethics committee in Italy because I just wanted to get involved in that process. So I sent my CV and my application to them, and they accepted me. Next slide. Safety here and comfort for trial participants are really important, and also trying to identify those studies which are not that sound or not that very well conducted, so trying to assess these issues. And the challenge is that often in an ethics committee you are the one the one patient trying to explain the needs of trial participants to researchers and clinicians who are considering them just as nothing important. Next slide, please. So the trial steering committee is a group of people who supervise the trial. They monitor the progress. They do not usually about defining the processes, but trying to make all the processes are in line with what is stated into the protocols. Some trials have for the community of patients. For example, in the HIV field, in the partner study, have a steering committee made up of 7 people, and 2 of them are patient representatives. So they are very well represented. You interact at the same level or other members to be sure that the trial procedures are conducted in a good way in respect of the trial protocol. Next slide, please. So in this setting, you can intervene very timely to where something happened. You can try to modify the trial if there is some unexpected result and so on, and also try to ensure that the clinical research is conducted well. The challenge is that you need to, there are frequent teleconferences, and sometimes there are some issues that need to be addressed very rapidly, very quickly. So you must be always up to date with the information coming from the trial. You must read all the documents of the communication about the trials. Next slide. Now, let's talk a bit about Data and Safety Monitoring Committee. I have no experience in this field, but you know that DSMB are a group of independent experts who can see the trial data before anyone else, before the participants, before the investigators themselves, and also before the sponsor even. So they can also have access to unblinded data during the blinded so that they can effectively monitor the safety of trial participants. So if anything unexpected or more or less unnecessary happen, they can issue a statement, a recommendation to the trial steering committee, just to say stop the randomization or unblind the trial or any recommendation they consider important for trial participant safety and also the quality of the trial itself. The inclusion of patient representatives is still poor in this group. We at EATG advocate for patients to be included in DSMBs, but this not always happen. Next slide, please. So the opportunity, of course, is to know the trial data before anyone else for the sake of trial participants, not just out of curiosity, of course. And so being able to issue an alert when something goes wrong there. But this is probably one of the most challenging activity you can do as patient representatives when it comes to the knowledge you must have. So you must have a good knowledge in statistics, because all the analysis of the data will be done, of course, using a statistical analysis, and also reading all the information and the documentation. Next slide, please. Health technology assessment, as Tamás briefly mentioned, is a relatively new activity which aims to evaluate, to assess the value of a new medicine or intervention or treatment or prevention strategy and so on. Usually the result of the health technology assessment is a cost effectiveness analysis. So a way to say this is an intervention which is worth spending an X amount of money or something like that. Next slide, please. This is quite important. It is pretty clear from Tamás' presentation that health technology assessment comes after the medicine has been developed. So often, usually it may be after even the marketing of the medicine or in parallel with marketing authorization application. So the scope is to have a say, or the opportunity for a patient to be there is to have a say about how big is the need for that new medicine, that new intervention for the community of patients. And so interacting with decision makers and payers, you can try to identify the best way to introduce that technology in the market. And again, here, usually individual patients are invited to this process, and the challenging thing is that those individuals should represent the whole community. Next slide, please. Let's close with the regulatory affairs. I've been a member of the Patient Consumer Working Party at the EMA for a few years now. In general, at the EMA, there is a very well established and articulated representativeness of patients within their activities. This is not the case, for example, for the national competent authority in Italy. So I cannot have the same level of interaction with at the national level. Next slide, please. You can see here a number of activities going on at EMA where the patient involvement is usually requested, and it comes from the opinion, the summaries of opinion for the pre-submission. So when a new medicine is assessed, they can have a conversation with patients. They can also invite patients to a meeting to explain their point of view. There is a package leaflet review, also risk management, internal management level, and also for safe communication. Next slide, please. So I think that the opportunities here is to be able to address the regulatory issues, but regulatory agencies are really highly regulated, so to say. They are really bureaucratic. At least the EMA, it can take months to understand what are the various committees and working groups and all the departments, how they interact, who is the right person to speak to if you have a specific problem, and so on. So there are really, and also there are some processes to be put in place to interact with some departments at the regulatory agencies because of the whole confidentiality, commercially confidential information they must deal with. The preparation of the meetings can be time-consuming, and also, yes, as I said, if you want to bring out a new activity, that must be carefully planned, and you must identify the right person to be speaking to. So I try to address very quickly, next slide, please, which is my last slide, and the thank you slide, all these issues of patient involvement. I don't know if there is any time for questions, but I'm here happy to reply if possible.
Thank you very much, Tamás and Giulio, for the very interesting presentations. I don't see any written questions so far. It shows how well you have explained everything. So, but if there are any questions, dear participants, please write them now, or you can also raise your hand, and then I will unmute you. This is not the case. So yeah, then I would like to
just one suggestion, that because as I said, we have recorded this webinar, and we will also upload it to the EATG and to the EUPATI website. So if you have any later questions that pop up, then I think you can just confidently write to any of us three, Christian, Giulio, or myself, and then we are happy to answer those questions that come up later.
Yeah, thank you. It's very true. So I wish you all a good evening, and thank you very much, Tamás and Giulio, for your presentations, and thank you, Christian, for your attention and your interest. Bye.
Thank you, everyone. Thank you. Bye. Bye now.